everyone, and hello Bill. Thank you for joining me today as we go down the rabbit hole and look at some of the niche aspects of genomics. Today we're going to be talking about all things cloning and of course Dolly the Sheep, which Bill was involved in. But before we start, Bill, if you could just introduce yourself and give us a little bit about your background.
Well, my name is Bill Ritchie, and I probably came to science by a slightly convoluted route. I did agriculture, a very practical course at agriculture. I then had the opportunity to do an Open University course, and at that time I worked for what was the Animal Breeding Research Organisation, which developed eventually into the Roslin Institute. And when my job on the farm, the Stanhope farm down in the Scottish Borders finished, I moved to Roslin, where I did large animal anesthesia for a few years. And then because the equipment became available and I showed an interest, I started micromanipulating embryos, and it's kind of developed from there. And I did eventually get a PhD by publication, and that was actually in Hungary. And recently I became a fellow of the Royal Society of Biology. So quite an interesting kind of route into science, and that was certainly the way that things developed. And because I probably had lots of these skills, then I was probably the ideal candidate for doing micromanipulation. A little bit of engineering skill, a little bit of science, and the two came together, as well as good hand-eye coordination through various sports that I'd played in the past.
Yeah, so your skills were kind of like very suited to kind of the cloning field. But would you be able to just kind of give us a little bit of a background kind of about the kind of cloning field? Because I feel like obviously the most prominent thing in everyone's mind would probably be Dolly, but there was also things done before Dolly. Would you be able to kind of go through that and the kind of ones that you were involved in?
When I got the equipment, it was very basic. I took over from a PhD student called Lawrence Smith, who still works in science, and that equipment became available. And the real problems at that time were that there were no good culture methods for sheep embryos. You could culture mouse or these sort of embryos. The culture for these was very well worked to quite a good number of embryos, but in sheep we could only culture to very basic levels. And the other real problem was that the glass microtools which were used to micromanipulate these embryos just were not available commercially. So the idea was that I had to first learn how to use tools to actually make the very small glass microtools before I could actually even do any micromanipulation. And all these things took maybe a day, at least a day out of the week, to actually make any basic culture medium, to actually make the tools. And the other big problem at that time was that there was no way of in vitro maturation of oocytes. So the very material that we were making had to be recovered from live animals. So that involved superovulation. And this work obviously went on during the winter when sheep actually are mating. So we could only work during the winter months, and we could hopefully get a reasonable number of oocytes from the animals by superovulation. But that involved some surgery as well.
So why was it that sheep were chosen? Why not like a rat or something?
Yeah,
what were the challenges as well? Kind of working with those.
it always seems to be counterproductive to use a fairly large animal. But there are some technical reasons. The maternal zygotic transition of the embryo occurs in the mouse at the 2-cell stage, and it occurs in the sheep at somewhere around the 8-cell stage. So it was thought that perhaps we would have that little extra bit of time when the oocyte or embryo was more malleable, and we could actually use that to be more successful. It proved that we could use embryos and then cells to a much greater degree later on, but certainly in those early days, that was one of the reasons. And the other reason was that we knew how to work with these animals. We'd worked with sheep for a long time. A lot of the experiments we had carried out were on sheep embryos. So if we wanted to do anything useful with the embryos in the future, then we had to be able to work with a larger animal than the mouse. It's not easy taking, say, things like milk samples from mice, although you can do it. So if you're going to use something on a commercial basis, then if you want to take milk, for instance, then you would probably need to use something bigger than the mouse.
But what were those kind of challenges with using sheep? Because obviously I don't really hear many people like kind of working on sheep now. It usually is just kind of mice.
Well, yeah, but you know, the mouse oocyte is around 90 microns, whereas the sheep oocyte, almost every other oocyte, is a little bit bigger, maybe up to 150 microns. So there is a technical reason there as well. And the mouse, the oocyte is very much more difficult to work with. The zona pellucida is very, very flexible in the mouse. So quite actually difficult to get through the zona pellucida. And people used various mechanical means to actually get through the zona pellucida when mouse cloning actually took off a few years after Dolly. So there are kind of technical reasons as well as size and you know, the reasons that I've already talked about. So yeah, there are lots of reasons, and of course, once you've actually done the cloning work, what you do is you can stick it out in the field somewhere, and it looks after itself. Whereas mice are a little bit more difficult to look after, and so you need specialist facilities for the mice. Not quite so much for the sheep.
But were there any challenges associated with working with sheep? Because I assume that they take longer to
They're bigger, yes. Yeah, I mean, you know, physically moving sheep is of course one problem, and because we were superovulating, then these animals had to be caught on a regular basis and given injections. At that time, the hormones that we were using were at a very short half-life, and it meant that a lot of the animals had to be injected twice per day with these hormones. So actually catching and handling animals was one of the problems. You know, I'll say this all the way through any interview: you're only as good in these experiments as the weakest link. The person who perhaps is not you know, doing their job would be a problem in any of the parts of these experiments. So they're very much a collaborative work between people producing cells, the people at that time we were using glass dishes, and all of those had to be washed and sterilized, and all sorts of things that people really don't think of. It was a really, really difficult experiment to actually carry out, certainly the Dolly one.
Yeah. So Megan and Morag, they were the first successfully cloned kind of animal from differentiated cells. So how did that kind of pave the way? Because I feel like there's Megan and Morag, and then there's Polly and Molly, and then there's just so many names. Like, what was the difference between all of those in terms of the technique used and stuff like that? And then how did that pave the way for Dolly?
Well, my boss always said that Morag and Megan were the enabling technologies, if you like, to actually allow the cloning to progress and for things to actually work. And the kind of reason for that was that the first animals that certainly I made, and there were quite a number of animals made prior to Dolly, but the technique, of course, was to take an embryo which had developed to maybe the 8-cell stage. So the cells are fairly easily disaggregated. So you can take one big cell containing a nucleus and place it into an enucleated oocyte. So an oocyte where you've actually removed the DNA. And at this stage, one of the problems is you can't see it because it's not bound by a nucleus in the oocyte. So removing it is one of the big problems. But you then had perhaps 8, maybe up to 16 cells that you could easily disaggregate and actually use for cloning. But cloning has a success rate somewhere in maybe 10%. So even if you had 16 cells, the possibility of making more than one clone is very, very small. So the animals prior to Morag and Megan were made by this method, whereas Morag and Megan were made from cultured cells. So the fact that you culture cells mean that you have many, many cells, so literally millions of cells, and then you can think about being able to genetically modify those cells. So you can actually have a cell line of cells which are all genetically identical. So you can make a number of animals which are all going to be the same, which are all going to produce the same chemicals, perhaps, that you're looking at, or proteins that you're looking at, rather. So that's really the big breakthrough in being able to go from that very small number of identical cells to millions of cells, and potentially being able to genetically modify those cells and produce animals which could have an additional gene at that time, or even knock out a gene. And we did both of those in subsequent years.
So what was the kind of difference between obviously Polly and Molly and then Dolly? Because I think I read that
it's Morag and Megan were the first ones.
Yeah, but then after,
and then Dolly. So I mean, what we'd used was a cultured inner cell mass from blastocysts to actually produce the first animals from cultured cells. And then, the question is, well, how do you develop that? Can you use a fetal cells, for instance? And it developed from there that we use, although it's not generally recognized, we actually use 3 different cell lines for the Dolly idea. And so we had some very early cells in development, some from a fetus, and then we had the adult cells. And the fact that we could, we were interested in using those techniques to actually make animals which had an added protein, a human protein. So these were all steps in the development to actually do that.
So with Polly and Molly, I read that they use somatic cells, but they also used they inserted like a transgene. I don't feel that. How did that differ from Dolly? Because I don't feel like that received as much attention as Dolly kind of has. So
Well,
what kind of differed between those two?
yeah, I mean, I think the idea of Dolly really just caught the imagination of not only science but really the whole world, and people then had the idea, well, maybe we can't clone ourselves. Maybe one of those things that people in the back of their mind think that perhaps this is just one of those things that they can connect with science, and they can see something in their life that perhaps would be affected by it.
Yeah. What was it like kind of at the time when it kind of like got like published and everyone became kind of aware of it? What was it like kind of from your
Well,
perspective? Because obviously you were a part of it.
well, in lots of ways, then the Morag and Megan paper was around 25 years ago, and problem was that it was dominated by other stories in the press at the time, so it really didn't kind of reach its full potential. But when the Dolly paper was going to be published, the story was broken early by some of the Sunday newspapers, and that, of course, caused the problem, because we really weren't we weren't we weren't really suitably sort of there at the time to actually get things actually working, and the press were not going to be there for 2-3 days until the paper had been produced. But because the story had been broken early, then all hell had broken loose well before the time that we expected it to be, and not that we really anticipated that it would be just quite such a big story. Certainly, I was beginning to get phone calls from my boss, Serene Wilmot, on the probably the Saturday evening, that the story was going to be broken early on the Sundays. Sunday newspapers knew about it. So he had already started kind of fielding calls from other people, and was going to go into the Roslin Institute to actually start dealing with some of the press. And I think he did a small number perhaps on the Sunday, but by the Monday, as I say, all hell had broken loose. I was still, busy doing cloning work at the time, and I entered the institute, and the car park was full of vans with with dishes on the roof, just trying to get a story there. And many of these people had actually flown in already from the United States and all around the world. It was bedlam.
Did you have any people like protesting outside?
Not for some time. People did kind of catch on. At that time, there were some problems, certainly in England, certainly around about Cambridge and Oxford, with protesters, but we didn't have a huge amount of problem with them. I think it was too far for them to travel. But we did have on occasions.
I think what's interesting about Dolly is that like I wasn't even born when Dolly first got cloned, and so many animals have kind of been cloned since. Like last year, I think like a black-footed bearer had been cloned or something like that. But why is it that in like kind of textbooks and teaching and everything like that, Dolly is kind of still in everyone's mind? Like if I said cloning to someone, I'm sure that Dolly the sheep would pop up in their head. Like that would be the first thing that they would think of. Why do you think that his kind of transcended like time essentially?
Yeah, I mean it is interesting, isn't it, that people remember this one scientific experiment, and in actual fact, it was rated higher than the first Mars landing in that year, just because of its significance in human medicine and stem cells and how things have developed and treatment for diseases, and just the fact that we had changed a scientific law. The fact that when I was at agricultural college, excuse me, the law was that once a cell had been differentiated, it couldn't be undifferentiated. And I can remember lots of people who taught science speaking to them and them saying, "Oh, it's terrible. I have to change all of my lecture notes because what I've been teaching people all of my life has changed." And it was that sort of change. And I think the fact that so many people did see it, I think if it had been a mouse, then no one would have paid a terrific amount of attention. I think what happened too was that one of my colleagues jokes about where the cells came from, and calling her Dolly also helped the process of making it very memorable for people who maybe knew about Dolly Parton and connected the two, and Dolly herself knows about it, and is fairly, I think, fairly happy with the fact that she has been named after her.
I mean, I think a lot of people kind of think what is the point of cloning? Like, I was looking kind of down the list of all the animals that have been cloned, and I saw that like a cat had been cloned. I don't know. So like sometimes you might think like, why don't you just go down to like the shelter and get a cat if you want a cat? But like, what is kind of the goal of cloning? Like, how can it be applied, especially with animals like that, like a cat? Like,
yeah, well,
purpose.
Because we have home office regulations in this country, the chances of actually cloning something on a commercial basis are fairly slim. It's not something that we would perhaps do very much in this country. People have cloned all sorts of pets. People always think that if they clone a particular animal, then the subsequent animal, the clone, is going to be a clone of that animal. And unfortunately, what they don't realize is that the original animal and cloned animal are can be really different because they are exposed to different diseases, different stimuli throughout their lives. If they're treated differently, then they're going to be totally different. We're all the way that we develop and the way that animals develop are more on the environment than their actual genetics, and people really don't understand that.
Yeah, I mean, what are some of the kind of applications of cloning? Like, what could it be used for? I know, like, kind of like in the medical field, like I know, like in terms of like conservation and stuff, like how can cloning be used?
Yeah, I mean, the original idea was to make animals which had a human protein. And at that time, 25 years ago, the problem was that proteins were often extracted from blood, things like blood factors for hemophiliacs. And the problem was that it was very difficult to detect viruses in the blood. So blood products were contaminated with HIV and other viral diseases. So sheep don't get these diseases. The idea of one of the commercial sponsors, PKL Therapeutics, was to actually make proteins from milk of sheep. And that was superseded by other methods. But this showed that that could be done. That we could actually get a flock of sheep which were identical, where we could produce milk with a protein, and that protein could then be purified, and then the person would not suffer from the same viral diseases as we had done in the past.
What about kind of going forward? Because obviously cloning is still done, and I feel it's mostly kind of done in the conservation sense, but also kind of people looking kind of for the like organ donation and stuff like that. Would you be able to kind of discuss that a little bit?
Yeah, I mean, recently we've had the some animals which were chimeras, which were made with human cells and primate cells, and it's always seemed to come from China, but these experiments were done in the early 2000s, very similar experiments. The problems with trying to make organs, and what you can do is you can take an embryo, knock out a particular gene so that perhaps you would produce an animal which had a liver or pancreas or something which didn't work, and then you could add a human cell, and the human cell would, which is has that particular gene in it, would then make a pancreas, and it would be a human pancreas in that particular animal. And the problem is, of course, that although it makes the pancreas, all the other connective tissue, the blood vessels and everything still come from the animal that you've transferred these cells to. So although it all seems a good idea, the problem is that you still have a pancreas or a liver or whatever, which is not from that the cells that you want. It's not completely from those cells that you want. So yes, it's this has been an idea for a long time. So although it seems to have raised its head recently, it's not a new idea, and the problems probably still exist. I don't know.
What about the applications of cloning within kind of conservation? Because I know that's a really kind of prominent field as well.
Yeah, I mean, at one time I said that one of the animals that we could clone would be the wildcat, the Highland tiger, and it's probably one of the few animals that would be very easy to clone, because we have things like they hybridize with domestic cats. So the chances are that the oocytes from the domestic cat, you could actually use those for the wildcat. So you could actually produce animals that way. It'd be expensive, but it would be a way of actually increasing it. Problem with many of the other species is that, one, we possibly don't know an awful lot about the culture conditions for oocytes and embryos. We don't have the ability at the moment to actually make to harvest oocytes, to actually do this, and we don't have the facility to actually transfer them unless they are an animal which are very similar, like the wildcat, to another species. So it's these things have happened in the past. The mouflon, the wild sheep, had been cloned a long time ago. A few other animals, but the problem is getting access to suitable oocytes and suitable recipient animals, especially when there are very, very few, unless, as I say, there's a domestic species which is close enough for them to use the oocytes and surrogates for that. No zoo really wants you to take an animal away and say, we will try and transfer an embryo into this animal and see what happens.
I mean, what about humans? Because I feel like everyone, when they think about cloning, they think that they're going to get cloned in like in a few years or whatever. Their clone's going to be kind of walking around. But what are the challenges of cloning humans?
It's brilliant. It's one of those things where, as I indicated earlier, unless you actually know a little bit about the culture and all how to actually do things that way, then it can be difficult. Now, we do know quite a bit about human IVF, but the problem seems to be that for some reason that we don't understand, the success rate is very small. Experiments have gone on in human embryos for many years, and people have managed to work with them, but again, the supply of oocytes and embryos is very difficult to get, and human reproduction is pretty inefficient when you compare it with animals. So if you compare it with where perhaps 95% of sheep will be will get pregnant on the first. Well, there are obviously differences in that these most wild animals, most domestic animals have a period where they are receptive to fertilization. So within that period, something like 95% sheep. But in humans, it's probably the other way around about. We have no actual period where it's easy to detect when the female is receptive, and we have these other technical reasons that we really don't understand. So yeah, it's going to be pretty difficult, I think, to clone a human. The other, apart from ethical reasons, where we perhaps have too many people in the world, is I think it's just going to be very, very, very difficult to get that number of oocytes and embryos and recipients. And if we're not 100% sure that that baby that's produced is going to be fit and healthy, then another reason perhaps for not doing it.
How do you think kind of newer technology, so for example CRISPR, will kind of improve our ability to kind of clone? And how does that compare to kind of the techniques?
Well, it's like everything else, science never stands still, and the development of CRISPR-Cas9 is probably just one of the breakthroughs in science at the moment, where instead of having to actually insert a gene, clone from it, then we can perhaps do a very much more targeted approach to genetic modification or genetic changes in embryos. It's not something I know a huge amount about. It's I did most of my my cloning work quarter of a century ago, so it's quite difficult to keep up with everything.
Yeah, I was going to ask you, I know you're retired, but how do you think that the cloning field will evolve over the coming kind of few decades?
I think unless there's a big breakthrough in the success rate, I think that it's going to kind of retain its scientific side of things, rather than making lots of bets for people who've got too much money and don't really understand what they're doing. There have been some sniffer dogs which have been cloned, which and this seems to be a trait which is which actually is passed on through the process, the cloning process. But from the point of view of cloning wild animals, then it may not. It's a scientific technique more than anything else, and there may be developments in the future. I used to work with a little bit with some of the zoos, and one of the things that I said at the time was, well, we now have stem cells. If we can differentiate these stem cells into oocytes and sperm, then all we need to actually make an animal would be a sample of tissue. And from that, the developments of making stem cells from any type of tissue, more or less, then if you can make an oocyte and a sperm from there, then all you need is a surrogate to actually produce another animal. Quite a thought, really, isn't it? That you, in the future, you may only need a tissue sample to actually make another animal.
Yeah, I mean, the cloning field is amazing. I think everyone's kind of attention will be on it as like with time goes on. But thank you so much for joining me today. It's really been great talking to you. It's been really, really interesting. So thank you so much.
Well, thank you. It's nice to speak about some of the things that you've done in the past, and let's look forward to seeing what happens in the future and how the developments of 25 years ago are now going ahead and helping to treat human diseases.
Thank you.
Thank you.
Hello everyone, if you enjoyed this video, then make sure you check out some of our other videos in our series by subscribing below or going to our website, frontlinegenomics.com. I hope you enjoy.